Good night, good afternoon, good morning, wherever you are in the world today, and welcome to the first webinar of our AI Unbounded series. Thank you all so much for joining us today. We're really excited to launch these online events, which will be running on a monthly basis, so stay in tune with further announcements. A few notes for today's webinars. So as you've probably seen, everyone's microphones are going to be muted throughout. If you would like to ask a question, you can use the chat functionality on your GoToWebinar panel, and you can just send me a question directly. My name is Emma, or then just send a chat message to the rest of the group, and we'll have a short Q&A session at the end. So I'll now pass over to Lindsay Smith, our field application scientist, who will be presenting today.
Thank you, Emma, and thank you everyone for joining today. So first, we're going to start with a little bit of introduction, just very briefly, and then we're going to jump into the cloud and do a live demonstration of some of the features I'm showing here. I apologize if you hear anything in the background. That is my dog chewing on a bone. So hopefully she quiets down. But let's go ahead and get started for today. So today our theme today or of this month is speed. So what are these nice tools that we have to help speed up not only training the model, but getting to a functional AI model for your data analysis soonest. So we do want to start a background here. Let me go back into presenter mode. And so I just wanted to give you an idea of who all of our Aiforians are. So our Aiforia users really do exist around the world. There's 3,000 plus users in over 50 countries, and we have processed and analyzed over 1 million images with over 400 AI models. So there's quite a breadth of models that we've developed and that our customers have developed in several different uses. For instances, we have pathologists, medical scientists, researchers, and clinicians that all use Aiforia, not only for image management, but for AI model development. So I do want to pause and take a moment and kind of give a shout out to some recent publications that were accepted and or published. So here, the Finnish Institute of Molecular Medicine recently developed a low-cost digital microscope scanner, and this was built to be able to digitize blood smears in rural Tanzanian populations and apply deep learning algorithms developed by Aiforia to detect malaria. So this is a really nice new publication that just came out. In addition, this article was just accepted by Human Pathology and should be published in the January 2021 edition. And here, the Individualized Drug Therapy Research Program, University of Helsinki and Helsinki University Hospital, were associated in helping to utilize an AI model developed by Aiforia to quantitate fibroblast foci and inflammatory cells in idiopathic pulmonary fibrosis. So please stay tuned in next month's live webinar series, and we'll give more publication shout outs and spotlight some Aiforians and their AI models that they've been creating. So the Aiforia offerings really are trifold. We have our platform itself, our people, which is a huge portion of what we offer, and our deep learning AI, which you're going to see a bit of live today. So our platform is scalable, not only within your lab, in your department, but also across institutions. And we are 2D image agnostic, so we can handle multiplex immunofluorescence images, IHC, H&E, you name it really. And because of this, we're also scanner agnostic. So you'll never have to purchase a new equipment or purchase any new equipment really to be able to use our platform to build your AI models. And we have been secure and cloud-based since our inception in 2013. So we're one of the longest based AI model development platforms to date. And really our mission is to remove the most common barriers for researchers and clinicians in order to access AI as a research and diagnostic tool. So in that way, we've worked tirelessly to create an easy to use user interface. We really want to empower our Aiforians and our users to be their own experts and to really be able to creatively and flexibly build these AI models. And we're always adding new features, which we will in very short 30-minute and once a month we'll display the new features and up and coming latest greatest tools and tricks in this AI Unbounded series. So our people, we do have dedicated onboarding specialists, but our whole team really is made up of scientists and software developers and engineers. So in that way, you don't need to know how to code. You don't have to be a data scientist to be able to use our platform. You don't even need a dedicated computer, actually. You don't need your own dedicated GPUs. We provide all of that for you. And so our science specialists do range from vet pathologists to cancer biologists to microbiologists, neuroscientists, engineers, etc. We do have an in-house annotation team, so in the event that you don't have the time to build your own AI model, you can outsource that to us. And then we definitely have additional ongoing single point of contact support. So as far as your deep learning AI that you can build, it's really as simple as labeling your input data, training the neural net, and then you deploy your your AI for image analysis. You can easily update these neural nets, which you'll see later with transfer learning. You'll be able to mix and match them. If you, let's say you have an additional question that's come up, but you have two different AI models that you've built and you want to merge those, you can now do that. And because we are cloud-based, this means that you can collaborate not only locally, but globally. And again, we do have industry-leading tools like our image match, which we'll show in a later webinar, our Annotation Assistant, which is our patented active learning tool, which you'll see today, as well as our external validation. So the way that this just quickly works is and so before we really jump into the cloud, I wanted to go over how this works and a few sneak peek examples. And so, but the general workflow here is you take any 2D image, you upload it with any scanner. Once the image is uploaded to the cloud, you can then develop your own AI model using Aiforia Create. And then, or let's say you don't have the time, you can deploy that to get a bespoke model that we built for you. So what's really nice is that neural nets do not get tired. They are highly accurate. So you can automate these manual tasks that take up a lot of human time. And this really allows you to standardize your image analysis, go much more quickly than you would have been able to do before. And again, it's really nice that you can collaborate remotely. You don't have to physically be face-to-face with someone, especially now with the global pandemic. Fingers crossed for the vaccine availability soon. So here, I just wanted to give you a sneak peek of some of our tools that we've built in order to expedite your workflow. So first up, we have our analysis to annotation converter tool. And so essentially what this does is if your AI is performing really, really well on a portion of your image that has not received any annotations previously, but you still know that you need to increase that annotation number to really bulk it up, you can now convert that that analysis into annotations to be used in future trainings. And so I'm going to activate these two videos simultaneously, and it is quite a fast tool. On the left, you can see a single cell class in red where about 100 objects are simultaneously converted into annotations. On the right, you see the tool also works when you have more than one type of object, so multiple classes. And this is great, so it works on object detectors, but what about segmentations of entire areas of tissue or subregions? So here on the left, you can see a multiplex immunofluorescent images where we're actually trying to segment out the soma, the somatic, and the nuclear compartments. On the right, you're seeing a nice whole slide image of a cerebellar slice where two different tissue subregions are being segmented. So you can convert very complex semantic segmentations. That's the term for regions when you want to get a total area of something. So you can convert that nicely and quickly, and there really are no ROI limitations. None of these videos have been sped up. These were all shot in real time, and they're playing in real time. So lastly, what I'm going to go into a little bit more detail today and show you additionally is our patented active learning tool. It's called our Annotation Assistant. And the whole purpose, and I apologize, that is my dog. The whole purpose of your Annotation Assistant is to really assist you. It scans your whole slide image, and it shows you where the neural net is struggling the most. And it suggests annotations for you, and it asks you to update it. So it's really like, it's like getting rid of those weakest links and improving your model even faster. So instead of you having to go through and try and figure out where it's struggling, it automatically does this for you. So I'm going to turn this on. It works for your area segmentations as well as your object detectors. You'll see that as it goes through, it makes suggested annotations, which you can accept. But then we also have a multifunctional tool that, let's say you don't quite like what it suggested and you want to update that annotation and improve it before you put it into the neural net, you can go ahead and update those annotations with the tools on the right. And then once you've now done that, now all of these assisted annotations go into your training data for the next round of training of the neural network. So with that, we can actually switch over to the live portion of our platform. So here we're actually within the cloud. And what we're looking at is we're inside of, we're looking at that cerebellar, that whole slide cerebellar slice that we saw earlier. It is a multi-layer, multi-class model. Here we have our feature tree. And let's just kind of look at what this means. So if we just look at the analysis results here, we see that we have several features. They all turn on at once. Let's kind of toggle things on and off so we can look at them individually. So here what we wanted to do is we wanted to segment out in turquoise the cell body layer. And then we call that the Purkinje layer. And then in the red, we segment out the molecular layer. And you see here that we've actually specified that this AI model should only detect high quality in focus tissue, because as soon as this layer was out of focus, it made labeling the ground truth for these different cell populations quite difficult and almost impossible. So to be conservative in our interpretation of the data, and as part of a QC step, we only included high quality tissue in this AI. So we can actually toggle those on and off individually. I'm going to turn those off, and let's look at some of different cell populations, specifically within that turquoise Purkinje layer here. So within the Purkinje layer, I'm trying to identify 3 different cell populations. We have 2 cells that stain with the same stain and have traditionally been thought to be the same, but we now see that they have a slightly different staining pattern. Some of them are completely filled in dark here, and then some of them are sort of hollow looking and empty. And so we're now beginning to discover this investigator is actually looking into the function behind these different cell populations using electrophysiology, but they also wanted to know, is the distribution pattern of these different in the cerebellum within the slice? So consistently throughout the different bregma levels of the cerebellum, they did make this finding. They found that these darker cells, these more filled cells, and you can see by the heat map, they more localize laterally and towards the outside of the slice, whereas that sort of empty cell population that stains with the same stain, it's more central and sort of towards the inside of the lobules, which was really interesting finding. The interneurons then, how are those localized? And what was interesting was those were kind of ubiquitous across all portions of the Purkinje layer. Next, you also have, we wanted to say in this red region, the molecular layer, we wanted to count all of the interneurons as well. And you can see there's quite a few of them. So let's say, let's just kind of review one of those tools that we saw earlier. That is our converter tool. So I think that I only have just a couple of annotations on this slide here and here, but let's say I know that I need more annotations. I need to give it, the neural net, more examples so that it can generalize across all of my slides in my data set. Well, one way that we can do that really quickly is just use our converter tool here, circle the region that you want to convert, and very quickly you see all of those analysis results have now been turned into annotations. There's a lot of keyboard shortcuts here. I just press the A button to turn on and off the analysis. If you make a mistake, you can always press Z, and that's going to undo your most recent move. Again, there are no ROI limitations on this, and so what's really nice is that you can easily add hundreds of annotations if you need. It also works on your segmentation regions. So let's say you really like the analysis results here. You select the layer that you want to convert. Here, you draw over that region, and then that is nicely converted. The last thing that you would then need to do is just add a training region. The purpose of the training region really is guides the neural net to where to look to learn on your new features. So let's go back maybe to this Purkinje layer cell populations. We have 3 populations here. Let's say that you're annotating along and you've added annotations here, for instance, and you realize maybe you get a few annotations in, you realize, oh darn, I have accidentally selected the wrong class when I did that. We now have a tool that's really nice. You just right click and you can immediately change the cell subclass. One thing though that you'll notice is now these cell sizes, or these object detectors, are a different size than the rest of them in this class. Let's go see what we want those to be. 15 micrometers. Instead of having to go delete these, for instance, instead of having to delete it and then redraw it by updating the size here, you can now batch edit all of the object detectors in that class across your image or in this image only all at once. And this is a really nice tool. You see it was immediately converted. So you'll never have to go back and sort of redo annotations because of small human error like that. So what happens when you have all of your annotations in, it's the latest, greatest model, or it's the latest, greatest annotations, and you want to go train it. So you do that here from our training tab. And when you train the model, you'll see that it says layers to train. And you see there's 3 layers here: tissue segmentation, the molecular layer, and the Purkinje layer. It's important to note here that each layer is a separate an individual neural network. So your AI project, what we're within right now, within the cloud, we're building your AI model within what we call AI projects. This means that your AI project is a multi-layered neural network. So here I have a tissue segmentation layer with 2 classes, the Purkinje layer and the molecular layer, as you saw before, which were in the turquoise and the red. So the entire job of this neural network is to segment out the Purkinje layer and the molecular layer. And as you remember, within the molecular layer, we had within the Purkinje layer, we had 3 cell classes, and the molecular layer, we had 1. What's nice about this, having separate individual neural networks, is let's say I'm working on different versions of this model, or I'm advancing the model, and I hit version 4 or version 5, and the tissue segmentation looks great. I don't want to have to train it anymore. I don't want to have to waste time training that neural net again. What's nice is you can unselect this as a layer to train. You can and then you can choose to only train those layers that have the object detectors. And then once you've unselected that, you can either choose a previous model that you've created within this AI project. So let's say I want to preserve how well it learned in this last training, and I don't want to have to retrain it again. Additionally, let's say you want to bring in what you've trained in a separate neural net on tissue segmentation on cerebellar slices, and you actually want to use an external net here. So you can technically bring in a different model or different neural net into this AI project, and you can directly apply it to these images. And so this is called using an external AI model, and here this is directly using an external net. So it will ignore all of your annotations in this project. Let's say you didn't want to have to re-annotate this and you know that it's going to function well, you can just go ahead and ignore and use it. What if you are updating a model? What if your protocol kind of changed a little bit and you know that the staining pattern is slightly different? What you can then do is now use transfer learning. So you can go and you can select the model that worked. Here I can just do generic tissue. You can select the model that you know worked, and then let's say you've made maybe 20 to 50 annotations, and you want the neural net to learn 50% on those annotations, then you would retain 50% of what the previous neural network learned. What if you want to retain more than that? Let's say 85, and you only want to retrain on the remaining on these annotations that you made. This is a quick and easy way to combine neural networks to update them easily in the event that, say, protocol changes, slicing patterns or slicing thickness changes, or maybe you just want to get really creative with your neural nets and mix and match things that you've already made. So that's one way to do it here. You can either directly bring in an external net, or you can use transfer learning. Okay, so that's a really interesting, nice new tool that we have. So again, each individual layer you can do this. Let's say I want to simultaneously reuse a layer that I've previously made here, but then for that second layer, the molecular layer cell classes, which includes the interneurons, let's say I want to use an external net for that. You can do that. You can select your external net here. It'll ask you which class you want to add that to or which layer you want to add it to. You just click OK. It'll ask you to update it here, and that is going to work quite nicely. So then you have your Purkinje layer. Let's say you only want to train on those cells. So here what we've done is in one single training, we're going to train one layer on a previously trained neural network. The second layer, we're going to use an external model, and the last layer will train completely new here. Your iterations are one complete forward and backwards pass through the convolutional neural network. So the more iterations you run, the deeper the training. You'll be able to get your verification. This will tell you how well did the AI learn against the annotations that you gave it here to learn on. Analysis allows you to analyze how is the AI doing in regions that were never annotated. how did it, so it takes what it learned on the regions that you gave it to learn and it applies it to a part of the image that it's never seen before. And then you can also prepare the annotation assistant here. So what that's going to do is that automatically does a whole slide image analysis, and that's going to take you to where the neural net is struggling the most. So let's take a quick look at that. With your annotation assistant, it will scan through all the images and it will make suggested annotations here. You can toggle on and off your annotations with the Q button. And then you can kind of say, well, maybe you weren't sure about that. You can use this multifunctional tool. They're the same tools that you have. I want to reduce this training region because I'm just not sure if that is a cell or not. But if you are positive about the cell, you can go here. You can add, and now, as soon as I accept this, this now goes into the neural net for further training. So it works. If it looks great, you can go ahead and accept it. Maybe you need to add one more cell type. Ignoring a suggested annotation by the annotation assistant does not in any way change the next suggestion that it's going to give you. What it's just simply ignore. So as this is going through, it starts at the area, it starts with the suggested region that it's struggling with the most, and then that sort of incrementally goes down, so lowering an error rate along the way. Again, this works for all layers in the project. You can do it for your semantic segmentation, your area region segmentations, and you can also do it for the object detectors as you saw. It's really easy just to quickly update those and then accept that in to the neural net. So as soon as you accept it, so this one looks great to me, almost. There's part of the molecular layer here that's missing. So I'm going to go ahead and draw this. It can be really quick. It doesn't have to be perfect. The neural net will learn. So we just continue sort of in that fashion until we know that we've added in enough in the region. So this, a blank training region, one important note about a training region is it has two jobs. The first job is this is the ROI where the neural net goes to learn on what you want to annotate. So if it's, but the second job is anything that is unannotated in a training region indicates background or artifact. So what it's doing here is it's appropriately found the glass slide as background. That's why there's no colored annotation in here. So you can go ahead and say accept. So maybe it was struggling at this border region on the slide between where the image ends and the glass slide starts, and you want to further define that as background. You just go ahead and click OK. So lastly, let's say you have your trained model and you're ready to move into the next phase of the process, which is you need to validate your AI model before you deploy it for image analysis. So how do you do that? Number one, people often do compare semi-automated counts and things like that to the AI model performance, but we also have a special tool built in specifically for this purpose. So let's say, so this is actually our validation tool. And so our validation tool here allows you to invite validators. We suggest at minimum 3. 3 to 5 is usually really nice. You invite experts, colleagues to come validate your model. You add their email, you send them an invitation. They get an email invite. In that invite, they will be asked to create their own secure login. And then once they log into Aiforia, they don't see anything about the rest of your subscription. They're directed to the AI project. They don't see any of the annotations that were used in building the AI, but all they have, and the identification of your slides are also coded so that they can't even see those as well. So it just converts into like a 1, 2, 3, 4, 5, 6 instead of the actual slide image names that you gave it. So once you have invited your validators and they enter this, what they're going to be directed to do is go to validation ROIs in order to provide annotations. And so you'll want to do this for each layer. So for the Purkinje layer and the molecular layer, the tissue segmentation, we'll draw this ROI here. Once that validator enters, you'll be able to see their names from a drop down here. But then all they have to do is simply go in and mark this. This was the molecular layer. Let's say I drew another validation region here. This would be the Purkinje layer. And then, oh, that's an ROI. So now I need to actually give it the validation annotation. And then lastly, it works, of course, for your object detector layer as well. Because it's a separate layer, these are separate validation ROIs. So you just take that ROI, once you click on the that second layer down, the Purkinje layer. And what's nice about this is you've seen now, we've nested this in our feature tree in a way that it's only going to find Purkinje cells and interneurons in the Purkinje layer, because the Purkinje layer is the parent. So it will only find these when it finds this proper cell layer, and it'll only find these purple interneuron classes when it finds molecular layer. So you can be pretty flexible in how you design this. You can draw this annotation here, and then you ask your validators to come in and label these appropriately, depending on how they view these. So they can change the size. That's less important here. So then they can easily click to multiple validation ROIs. So let's say we have another one here. They easily click here. They're immediately directed to where they need to annotate. And then as soon as they're done, once you have your latest, greatest model, you go to your AI model history, you open that up, and you press the validate button. Once that's done, you can actually export and analyze your validation results. You can then look at the inter-observer variability. How did all the humans versus the AI work? You get your false positive percent, your false negative percent, your total error rate, your F1 score, and your precision and sensitivity. So this is a really nice tool. With that, if there's anything today that you've seen that you want to see more of, you can always book a demo here on our website in the upper left. And with that, I just want to say thank you to everyone for your time, and I'm going to hand it back over to Emma for questions.
Great, thanks so much, Lindsay. So we have a few minutes left here, so we definitely have time for some questions. If we don't get around to everyone's questions, we'll email you with our answers afterwards. And as Lindsay said, we would definitely recommend that you book a demo if you want to see any of those features again, or if you'd just like to see an overview of the Aiforia software on your own images. But I'll get started here with the first question, and feel free to submit any more through that chat function and we'll pick those up. So Lindsay, the first question I have here is, how many color channels do you support for multi-channel immunofluorescent images?
Right, so we actually support many color channels, up to 50, I think you can get, but I can show you quickly. What's nice is that you can actually turn them on and off individually. So let me just show you real quick within this project. And I can show more of this in the next webinar if anyone's interested. Feel free, if you want to see specific things in these AI Unbounded series, go ahead and write into us, and we will incorporate that. We support up to 50, possibly more, but 50 I know for sure. Here, this is just 3 color channels. You can turn them on and off individually. I'm sorry, this webinar thing's in the way. And I just wanted to show you quickly. So here we can look at the MAP2 stain, which is the red. We can turn on the DAPI individually. And then we can also turn on all 3 channels. This last channel is TDP43. And the whole goal of this project, really, if you want to get an overview of the results, was to get this somatic and nuclear staining, not only of each individual cell here. So we used instant segmentation, which we can go over next time, but also to look at the somatic and nuclear compartment for each positively identified live cell. But hopefully that answers your question. We can support many color channels. You can optionally turn them on and off while you're annotating to help you better annotate and better view what you're doing. And let's, even if you can also choose which colors you want. So let's say you don't like the colors that they normally are, you can change the color of your channel if needed, or maybe it's hard for you to see the difference, then you can absolutely do that.
Great, thanks, Lindsay. So I'm conscious of time, so let's take on one more question. This one's a little more general. And the question is, typically, how many images will it take to train an AI model with Aiforia?
Right, so not many. So it takes about 10% of your entire, 10 to 20% of your entire data set you'll use. So let's say you have 100 images in whole slide images in your entire data set, about 20 of those would then be used to train your AI, and about 10 for validation. if you choose to go that route.
Great, thank you. And right on time. It is exactly half past. Well timed, Lindsay. So thanks so much for your presentation. And thank you so much to all the participants. We have been recording this session and we'll be sending around the video recording tomorrow. And please do explore the website for more resources. And yeah, thank you to everyone again.